Thank you. Thank you very much for having me today. And I'm very glad I will be able to attend this small meeting with you all discussing what I've been working on in the past few years. And today I will talk about how wastewater can be used to monitor AMR for public health and based on what we did in Saudi Arabia. So now AMR is often framed as a clinical issue, but it is equally an environmental transmission problem. And this image shows the Ganges River in India, which is a place of enormous cultural and social importance. But it also illustrates a key point that the large human activities can leave a measurable microbial and the AMR signatures in the river and the environment settings like waters. And the recent study focused on the mass gathering events in the Ganges River, and the study has shown a measurable sudden increase of certain ARGs in the river water after the mass gathering events. And it shows that water integrates signals from millions populations. And if we know how to interpret these signals, the water can tell us things that clinical case reports or individual patients testing that often cannot tell us in time. So in this framework, the water and the water sampling became a window for us to know the AMR for the public health dynamics. And among there are many different water types, the wastewater are able to capture signals from the whole populations, no matter they are symptomatic or not. And the wastewater are able to aggregate what the individuals shed and turns that into a community level samples and easily accessible to scientists working on AMR. And this is especially valuable to us is because the wastewater-based surveillance allow us to track the AMR at the scale where it actually circulates, and not only in small places like hospitals, but across the communities of a country, of a city, or potentially across the national borders. And operationally, when we collect the sewage, wastewater, such like sewage, we extract the nuclear acid, and use the sequencing technology and bioinformatic protocols to profile the AMR from the sample we collected. And Saudi Arabia gave us a particularly powerful setting in which to test these ideas. It's because this kingdom hosts the largest recurring mass gatherings in the world, so-called Hajj and Umrah, with millions of international travelers and domestic travelers, which can strain the public health planning and the response capacity to the public health emergency, in particular, something raised by AMR. And such MGs were believed to facilitate the faster and broader AMR transmission is because of the global attendees of such mass gatherings might be the carrier of ARGs and ARB. And from the view of a One Health framework, this might also become a challenge to the downstream wastewater treatment and also the water recycle and the environmental health. However, during the pandemic of COVID-19, like most countries, the Saudi Arabia closed its border, and that results in the restriction of the gathering. And after the middle of 2021, the border was reopened, and the attendance of such events is turning back to normal. And according to the data from the Saudi official, the attendance increased to 1 million for foreigners observed in the April and July of 2022. So we left with a few questions after this. So for the first time in the modern history, we were able to give an opportunity to observe this kind of mass gathering events under different number of attendees and the composition. So that's able to give us opportunity to witness the AMR in the presence and absence of such a large-scale mass gatherings, and also to assess the profound influence of how the mass gatherings will have effect on the local AMR. So to track this process, we collected wastewater from treatment plants in 2 cities in Saudi Arabia, which is responsible as the host of such mass gatherings. And we also have a control site, which is a semi-enclosed campus that was not affected by attendees of the mass gatherings. And we collected the wastewater from these 5 different points. And the sampling design will allow us to compare the period of the restricted travel with the period of high international attendance, and to ask not only whether the AMR changed, but also whether the specific imported AMR or ARBs could be detected. So, and that is exactly what we observed. And by comparing the metagenome from different time period, we found that newly identified ARGs starting from the reopening of the border. This is a graph showing all the detected ARGs from the beta-lactamase family across 5 different time periods, including the year of 2020, 2021, and 2022. And when we're comparing the detection level of different ARGs, we find out there are 4 ARGs that was never been found before the border reopened, which are the PER, YRC, FOX, and PFM, that it suggested a potential origin of these MBL or ESBL genes that might came from the outside of the Saudi Arabia of the kingdom that might be introduced by the foreign pilgrims after the reopening of such borders. And when we take the next step and move from the metagenomic detection to the isolation to the isolate level validation, we were able to find several bacteria that happen to carry the ARGs we found after the border reopened. And by conducting the ONT sequencing, we find out there are 3 different bacteria species, including the Shionella putrefacens in the pink color, the Aeromonas media, and the Shionella shamanensis. They all happen to carry these PER genes on their chromosome or the plasmid. And all these 9 cases are coming from the different time points of the wastewater sample, but they're all detected right after the border reopened. And more than that, we observed that the location of these PER genes are changing from chromosome to the plasmid that suggested a high potential of the horizontal gene transfer risk. So to further investigate the potential of horizontal gene transfer, we analyzed the flanking regions around the PER genes across the bacteria genomes it carry that. And take the Shionella genomes as example. The PER genes were found on the plasmid on 1 colony at the very top, and the chromosome of the 5 colonies of the chromosome, as well as the Shionella shamanensis also carry the PER on the chromosome as well. So the chromosomal PER resided within IS26 array, further detected to be a genomic island, and which included more than 3 ARG clusters in this specific region that confer resistance not just to the PER conferring the beta-lactam antibiotics. We also found the ARGs resistant to to other antibiotics, including the mycolide and tetracycline. And in particular, the emergence of the tetracycline, the TETC genes, suggested a critical period for the ARG acquisition between right after the national border reopened after the 2021. So in sum, this study looked into the ARG and ARB of the wastewater sewage during the different mass gathering events in the kingdom since the border reopening, and we found that the wastewater-based civilians are able to provide different information by review the dynamic of AMR during MGs and indicating the varied response to the MGs. And we were able to detect and isolate the new ARGs which were introduced to the local wastewater system after the international level mass gathering events. And through the genomic analysis, we were able to find out the active horizontal gene transfer of particular genes that was happening right after the border reopened. That highlight the complexity of the AMR transmission pathways and underscoring the urgent need for more comprehensive strategies in combating AMR in global context. And with that, I would like to thank my lab, the members that helped with this project, and our collaborators from the Saudi University, and the funding is from the KAUST WDRC, and the work was published on Wastewater, and please feel free to reach us. Thank you very much.